All right, hello everybody. Welcome to the Let's Get Lit series, where we talk about interesting literature topics in the synthetic biology space. I'm Alan, and these are my friends Angel and Colin. And the way that this series works is, I guess we can do this weekly. Yeah. So every week we'll find some interesting synthetic biology topic or bioengineering topic, and then we'll teach it to each other blind. And so that way, hopefully you guys get to learn and we also get to hear some interesting stories about bioengineering. Excited?
Yeah, let's go.
All right, let's go. So this first topic is on protein computing. And so proteins are these complex biological machines composed of amino acids that fold into a really functionally important three-dimensional structure. And so it's a very complicated sort of machinery. But then in terms of computing, we oftentimes think about these ones and zeros like binary, in a computer, all these ones and zeros, they combine in certain ways and they use certain logics to compute certain things. And so this presentation is about how these two can be combined together. So protein computing. This is the paper that we're going to be talking about. It's called Programmable Protein Circuits in Living Cells. Have you seen this paper before?
No.
Okay, how about you, Angel?
No,
Okay, that's a good sign. Yeah, this is a science paper from 2018. And I think it's really interesting how they do the work here, because I think it's kind of like a puzzle where, have you done like truth tables before, like logic? Yeah. Tables, okay, yeah, how about you like and, or, those things?
Yeah.
So they do that not with ones and zeros, like just on a paper, but they do it with proteins. Yeah. Which is really cool. And they also have certain applications too, which is also pretty awesome. So 2018 science paper that we're going to be going over. This is the, let me see if I can actually get the pointer so that way we can, there we go, that's much better. This is the overall idea. These Pac-Man things are proteases, and proteases chop up proteins. Yep, exactly. The idea behind this paper is to use these proteases in certain ways to give it that kind of 1-0 logic.
So does each protease have a different function then? Or like
yes,
like
I would say so. You'll see an example later on. But no, I mean that's a really good question though, because each protease is a different enzyme. Right. And obviously enzymes have different specificities. And so by changing the amino acid sequences, you can change which sites each protease cuts. And so the good thing about this, it's not encoded in the nucleus too. So it's just all in the cytoplasm. Everything can be expressed, and it's all very sort of compactified in the cytoplasm. And this is the main sort of key takeaway, where you have two inputs and you have an output. And by the way, to your point, the proteases, they are different proteases coming from different organisms. And this also highlights sort of the basic research. I mean, if you think about, who cares about tobacco etch virus and hepatitis C virus proteases and their specificity? I mean, obviously it's important, but I think applying it in this instance is also an example of basic biology sort of supporting applied science. Yeah. The output, by the way, is a fluorescence or a fluorescent protein, a fluorescence probe. Are you familiar with that, Angel? Sorry. No, you're good. What's that? Are you familiar with what a fluorescent probe is?
Not fully. I've heard of it. I don't know if you'd like to explain a little bit more.
Yeah. So the idea behind the fluorescent probe is that cells can be made to turn different colors. And so in this case, if it happens to turn green, that means that you're expressing this green protein. And the idea behind the logic gate is you have these two inputs. So for example, you could have the yellow Pac-Man, or you can have the red Pac-Man, or you can have both, or you can have neither. And those two inputs can essentially combine in different ways through a logic gate that's engineered to either make the cell glow or not glow. That's the idea.
So these are all of these like experiments, like you said, like a puzzle. So like just choosing different Pac-Mans in the situation and then seeing what happens in the end.
Exactly.
Experiment. Okay.
Exactly. So you have these two Pac-Mans, and they also it's like a design puzzle. And you'll see, for example, here. Here's like one such puzzle that we can talk about. In fact, I'll go through this first one So I think that's quite interesting. So here are the key players in this puzzle. The Pac-Mans are the proteases, and they chop. They chop certain sequences. The yellow circles are where the proteases chop. So for example, the yellow circle is where the yellow Pac-Man chops. The red circle is where the red Pac-Man chops. The circle with a dot inside of it is called a degron. That's an amino acid sequence that essentially tells the cell to degrade the protein. Essentially just chop it up, get rid of it. These zigzags are essentially just things that help proteins adhere to each other. Honestly, not too important, but it's good for optimization in the paper. And lastly, the green protein is the fluorescence reporter. In other words, how do we tell if this thing is actually being expressed or not? We know because of the fluorescence. So if the cell turns green, we know that this thing is present.
I have a question.
Yeah.
So for the like leucine zippers,
Yes.
do they alter those out to get like different levels of expression? So it's like, oh, if I wanted tuned adherence, would I tune the leucine zipper? Or
By adherence, do you mean, what do you mean by adherence?
so I thought the leucine zipper was used to make sure like it would bind to the protein better or like find the protein better.
Yes, yes. So these two leucine zippers are essentially anti-parallel, and they can also adhere to each other. And so this is essentially a way to essentially make this dark blue zipper bind to this
I see.
light blue zipper.
I see. I see.
Yeah.
And those are not tuned. They don't tune them in the paper at all.
I think it is optimized.
Okay.
Yes. But the fun part is this. So
Sorry.
let's take a look at what happens if we don't add either of these. So if we don't have the yellow Pac-Man and we don't have the red Pac-Man, then that means that these circles aren't cut, right? So that means that the degron, the degrader signal, is still there. So what does the cell do then, Angel? Degrades it.
Yeah.
So will you see fluorescence or not?
I assume no.
No, right? So this will be a 0, right?
Yeah.
So 0, 0 input gives you a 0 output. What if you add in the yellow one, Colin? What do you think?
If you chopped at the yellow,
Right, if you add the yellow one, it'll cut the yellow one, it'll get rid of the degrader. So now this one is free to kind of float around by itself. So we will see a signal here. Angel, how about this one?
I think it'd be the same.
The same,
Like same number.
exactly, right? If you add the red one.
I just wonder if the fluorescent would be different since the yellow is full of that.
Oh, I see. So these are independent experiments. Yeah, yep. And so, for example, I can erase these two. In this experiment, you only have the red one, but not the yellow one.
I see.
Right, yeah. So in that case, only the red one gets cut, but same result. Right? I mean,
Yeah.
still no degrader, so therefore you will see fluorescence. And lastly, if you have both, Ellen, what are your thoughts?
so for us, even more.
Right,
Because
exactly,
there's a greater chance to cutting it off.
exactly. And if you think about what we've just done, you've essentially just created an orgate.
Yeah.
With this system, right? We only get fluorescence, the cell only turns green if both of the inputs are there, right? If one is there, fluorescent. If one is there, fluorescent. If both are there, it's also fluorescent. Only when neither is there do you get no fluorescence. So this is protein computing. It's using proteins as logic gates, which is really interesting.
First, I find it like, maybe I'm thinking this wrong, that just having like just yellow Pac-Man fluoresces the most. Why is
That
that?
is interesting.
Right?
Yeah, I'm not sure.
I would think if you had both of them, you increase your chances of like binding and getting rid of that one.
Totally. Yeah, I'm not sure. That's a really good point. And it's consistent too, right? Across 3 trials.
Yeah.
So that's a really good question. I'm not sure if the fact that having both of them causes adherence to each other,
Yeah, maybe like.
and that kind of
Yeah.
interferes.
Yeah.
Right, maybe. But then, I mean, that also, I mean, the red one also is even lower. So I'm not
Yeah.
sure.
Interesting. Yeah.
It's a good question. Yeah. But the main takeaway is, it's very clear if it's a 1 or 0, like
Yeah.
on or off. So that's the main takeaway. That's the orchid. And so I guess, Colin, I might task you with this one. This is the new puzzle.
New puzzle.
Yeah.
Oh,
So
geez.
0, 0, 0. If you have neither, what do you think will happen?
Yeah, neither. Well, it's gonna degrade.
Gonna do it, exactly.
Yeah,
So
yeah.
no signal. And Colin, how about if you have only the yellow Pac-Man?
You're gonna need both for this one. Right? Sorry. Yeah.
Yeah, no, you got it.
Yeah, because you have degrons on either side.
Right. If you chop here, there's still one on this side.
Yeah.
Right. So it's still no signal in this one.
No signal.
No signal. How about with both?
Signal.
Exactly.
And why did they add the leucine zipper onto that one? And not
I
the
think
other
it's
one?
just optimized. Yeah, so I
Oh.
think this is just to optimize the signal. for
If you go
future
back to
signal.
the last one, you can see, right, like the, use like, oh wait, no, the yellow, sorry. That's it, yeah, interesting. I don't know.
I'm not sure. Yeah, I think they designed these with the intent of maximizing the difference in signal.
I see.
I'm not sure exactly, the logic behind why they chose to put here and not on this side.
Mhm.
Yeah, that's a good question.
Yeah.
Yeah. But in essence, what gate would you call this? AND gates, exactly.
Yeah.
Right. So you only get the signal if both of the proteases are present.
Mhm.
All right, Angel, you ready?
Yeah.
This is the harder one.
Yeah.
We have a new player. This is the purple Pac-Man, another protease that cuts this purple circle. All right. So in this case, we still have the two inputs, but there is a third element that's kind of already endogenous inside the cell. It's already present. So I'll let you give it a shot.
All right, well, to me, it seems like another AND gate. Because in order to free the purple, to be able to cut on the right side, you would have to take away both red and orange. At least that's what I think.
Interesting. Let's try it this way. If we have neither one of these, in other words, this is not there, this is not there.
Yeah.
Right. That means that these circles remain.
Yeah,
And that means that the purple Pac-Man is still
locked, bind it
there.
to,
Right.
yeah,
So what would this do?
nothing. Like it
It's,
won't,
it's active. It's active.
oh, it's active right now.
It's active
Okay.
currently.
Oh, my bad, I was thinking the other way
All
around.
good.
If it's active right now, then it would cut the purple on the right side,
And
and then it would, fluoresce, like positive fluoresce.
it would fluoresce.
Yeah.
Right. Exactly. So that's on,
Okay.
right?
Yeah,
What if you add the yellow?
if you have the yellow, it just wouldn't have any more. Well, would it deactivate the purple?
Exactly. Right. If you cut here, the
Yeah.
purple halves are going to split apart.
Yeah,
So this is the design part, right? They designed these two components to only come together in the absence of yellow and red Pac-Man.
right.
But with the presence of the yellow Pac-Man, you cut the yellow circle and that dissociates the purple Pac-Man. So
It's
therefore,
same with the red.
same with the red, right? So therefore, would you expect to see fluorescence?
No,
No. Right, because there's no purple Pac-Man, the degrader signal stays on,
Yeah.
so no signal. And for the red one,
yeah, same,
how about with both?
same thing,
Same.
yeah,
Exactly. So this is a new kind of, combination of outputs, NOR, right? So this is the
Right.
idea, sorry?
Sorry, nothing.
Yeah, so you only get signal when none are present.
Mhm.
So this is really interesting, where they've designed this protease for this specific logic gate, which is really
Yeah.
cool.
Yeah.
Yeah. All right, Colin.
yeah, I
One
was
more,
gonna
one
say,
more.
technically you could keep building up.
Exactly.
Yeah. Alrighty. Oh, so it's active in that position, right?
What is active?
It's active when,
Yes.
yeah, okay, just making sure. So if nothing's there, then it'll degrade, or it won't degrade, it will cut and you have fluorescence.
Right.
Still fluorescence,
With the yellow, you will still have fluorescence.
yeah, because the red's intact,
Exactly.
yellow's intact, then on the next one, if red's present, you'll cut the right purple, but the left purple will be good, and so you'll still have fluorescence. And then both have to be present, or, yeah.
Exactly.
They're
It's
very
interesting.
clever, they're very
It's
clever.
Yeah.
interesting, right? Yeah. So it's like a logic puzzle where you design proteins with the specific objective of getting a logic gate out. So this is protein logic, protein computation.
Hmm.
So exactly right. On. You only get no fluorescence if you have both inputs. It's NAND.
Yeah.
The NAND logic.
Do you want to, so the NOR and the NAND, where does that come from?
I think it's not or and not and.
Right.
Yeah.
Oh, okay.
Yep.
That makes-
So not and. So therefore, no, it's the opposite of and.
Yeah.
Not or, opposite of or. Yeah. I won't bug you guys anymore with this, but thank you for indulging me on that. I mean, there are 4 other combinations. If you think about, the 2 inputs, right? They could either be both off, both on, or 1 on, right?
Mhm.
And then the 4 combinations, you can either have on or off. So there are 8 combinations in total. We went over 4 together. There are 4 other ones shown here. And you can see the way to design the proteins. I encourage all the listeners out there, by the way, to go through this yourself and just think through the logic in each case. But I think for now that's enough for kind of our game. But the main takeaway is they're able to produce all of these 8 logic gates. And if you think about computers, that's all they are. Right? I mean, computers are just ones and zeros combined in a certain way to produce a certain algorithm, right? And I mean, you can add certain numbers together by using a bunch of ands and ors and logic gates. So this is really cool. We're using biology, you're using proteins for computational purposes. So that's protein computing.
That's awesome.
Yeah.
Would this have an application of like, rather than have computer storage, you can have like DNA storage?
I think the world's your oyster.
Right?
Exactly. Yeah, DNA storage or protein storage.
Yeah,
Of course, this is not as scalable as ones and zeros. I mean, the fact that semiconductors can be on atomic scales is kind of already insane. But it's just cool to think about how biology can be repurposed for this thing as well.
yeah.
And it's good that you ask for application too, because I think as bioengineers, people are always thinking about how do we apply this for problems in the modern world. And one of those obviously is cancer, right? I mean, cancer, overgrowth of cells don't stop undergoing mitosis, and so this leads to a tumor. How can we use this protease system to solve certain cancer problems? And here's how the authors thought to do this. They thought, well, I mean, Colin, what's kind of the hardest part, do you think, about cancer therapy?
Making sure I think you have to kill all the cells, right? Because if you have one cell left, it's still going to proliferate and give cells cancer.
Yeah.
Yeah.
But also, there's a reason why chemotherapies are so toxic. Right?
Oh, sorry.
I mean,
it's non-specific, right?
Right.
You're killing everything.
Yeah.
around it.
Exactly. Even the good cells, exactly. So, I mean, you bring a really good point about the fact that we need to kill every single cancer cell, but we also want to minimize other damage, right? I mean, that's the hardest part. The fact that cancer cells arise from our own cell is kind of the bane, right? Where we have to look at the differences between the cancer cells and our own cells to get that kind of selectivity that we want. So here's what the authors thought about. They thought, well, cancer cells, they have certain signals that tell them to divide and divide and divide. So here are 2 examples of those signals, SOS and EGFR. These are very common signals that tell cancer cells divide, 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 divide. What if we could rewire those signals to cause death instead? Right?
Yeah.
It's like hijacking the cancer's telephone signal in the middle. And
So
so
those signals used for anything else in the body?
that's a really good point. Yeah. Yes. Yes, they are. So EGFR, SOS, they are present in normal cells. They're just overactive in
I see.
cancer cells. That's a good point though, right? We want to somehow inhibit that from happening in normal cells.
Certain level of activity.
Yeah.
In order to.
Exactly. There needs to be a threshold. And so the way we can do that, or the way the authors thought about doing that, is this. If there is this divide signal, SOS, this is kind of the biology behind these proteins. The SOS protein will bind to a RAS protein. And then the RAS protein will bind to an RBD protein. Again, the names aren't too important, but the main idea is we engineer RBD to have half of our proteins. We engineer RAS to have the other half of our proteins. So when SOS is there, when the divide signal is there, it reconstitutes the protease. In other words, when the divide signal is there, the Pac-Man arrives. Right? Okay, now what happens when the Pac-Man is there? When the Pac-Man arrives, this happens next. The authors also engineered a caspase protein. Caspases, are you familiar with caspases, Colin? No. How about you? Caspases are proteins that are enzymatically involved in apoptosis.
Okay. Yeah.
Which is why they can cause cell death. Apoptosis meaning cell suicide. So activating these caspases will cause them to go haywire. They go around chewing up parts of the cell and the cell just dies. So how do we selectively activate the caspase in the presence of this yellow Pac-Man? Well, just add that yellow circle there, right? In the presence of yellow Pac-Man, the yellow Pac-Man will cleave that site and activate the caspase, which will then lead to cell death. That's the logic behind this sequence of events.
So how do they introduce these like engineered cells into the body? essentially.
That's a good point. These are all engineered. I'll show this in another slide actually. But these are all encoded in DNA.
Right.
And so this is actually a gene therapy. I guess if this were to be implemented, it would be a gene therapy where you have DNA constructs encoding these pieces and you deliver the DNA into your cells.
And then that could be targeted, right? Because I know, yeah. Right. Oh,
To certain
so
locations.
yeah, like
Yes.
we're going to look for a cancer signal outside of the cell group in order to deliver.
Yes. yes.
And compared, I'm not sure if you know this, Shannon, compared to like normal, like cancer treatments. Like it's extremely important for like a cost. I don't know if you have an idea compared to like normal therapy. How does this compare in cost?
That's a good question. I'm not sure actually how far along this is in terms of like clinical development.
Okay,
But I do know that gene therapy is definitely in its infancy.
okay,
And so in terms of costs, I've I know that things like Luxturna and other gene therapies are crazy expensive.
Mm.
But I mean that kind of
Possibility.
the possibility is there, the imagination is there.
Mhm.
And with the advance of technology and industry, I think the prices will go down eventually to suitable levels. Yeah, but I think in any case, in any therapeutic, the beginning is always very expensive.
Yeah.
Yeah. But it is good to keep that in mind though, as we think about this. But going back to your point, Colin, in terms of the selectivity, do you see a problem with this?
It's not so very selective, no? Or
Like if the cell was not a cancer cell,
yeah,
what might happen?
get the signal and kill itself.
Right. Right. So this yellow Pac-Man might still spontaneously combine and cause cell death, which is what we don't want. And so you actually predicted this earlier by asking me about the selectivity question, because the authors thought, well, that's not good if there's a high background for killing. So they introduced the third Pac-Man.
Yeah.
The third Pac-Man. How does the third Pac-Man play into this? The signal causes cell death. But even a low signal, we don't want it to cause cell death, right? How do we do that? We do that by introducing this purple Pac-Man that inhibits the yellow and green. So by adding in this purple Pac-Man, it will stop the yellow and it will stop the green and prevent cell death. Unless there is above a threshold of the purple signal, right? If there is a lot of signal here, then that will cause a yellow to inhibit the purple and override that inhibition. And so here's the idea. They add this third component exactly to your point, to improve the selectivity. So they add this purple Pac-Man to cleave the caspase and prevent it from causing cell death in the absence of signal. And also, and they don't show it here, but the yellow Pac-Man also has a purple circle there. So what does that do in the presence of the purple Pac-Man?
It inhibits the creation of the orange background.
Exactly, right? If the purple is there, even if this forms spontaneously, it'll still cut. And so that'll be okay. Unless there's an overactivation of the divide signal. In that case, it will cause cell death. And so this is their engineering design. And again, you can see it uses a lot of the elements from that logic, right? We have, this cross inhibition talk. We have all these these protease recognition signals. So a lot of things are inspired from those logic gates. And what are the results? The results are, oh, and by the way, this is just the DNA construct. You can see they have the caspase and the protein components, and then it's all in one single DNA construct, right? So I mean, this design is very impressive. I think to get this to work is absolutely insane.
Wonder how they put it all in one construct.
One construct.
How did they do that?
Yeah. One construct, one promoter. One promoter,
And then
with multiple ribosomal skipping sites.
yeah,
So this is just, a beautiful, I think, example of
it's
DNA
crazy
construction.
design, yeah.
And so, I mean, all these components are engineered into this 6 kilobase construct.
Imagine how the cloning process would have been. Oh, pain.
Yeah.
That's cool.
But I think just absolutely beautiful design. And what's more, it works. It works. In co-cultures, in other words, if you mix together cancerous and non-cancerous cells, only the cancerous cells die. right? So it's selective in that regard. Of course, of course, not perfect, but yeah.
Sorry.
Yeah, I'll walk through this slide as well. So
Yeah.
in the previous slide, there was the full circuit, which is what I described. They also have a positive control circuit in which the SOS is always on. Or sorry, the RAS is always on. My bad. Okay, and so this is a positive control in which there's a mutation that causes this signal to always activate and cause the cell to die, essentially.
Yeah.
So on the y-axis here is the reduction index. In other words, how much of the cells die. Higher up means more death, basically. In the positive control circuit, when the RAS is always on, in other words, the cell is always seeing the signal, the cells all die.
Yeah.
Right? They all die, they all die, they all die, they all die. In the full circuit, when the cells are only going to cause that signal to appear in the presence of the either EGFR or SOS, the cells only die if those are present, right? The cells that aren't quote unquote cancerous in this model system don't die. They're okay. This one is a little bit of background, of course, but not perfect.
It would have been so cool. And maybe they did do this, but I feel like it'd be like too many trials. But if you got like a curve of like how much signal would you need, like a continuous signal until you activate or like activated enough of the yellow Pac-Man to prevent purple from preventing stuff.
Yes, actually, so I think I didn't include all the images here. I only included the images that I thought were interesting enough to kind of put on the slides. But they do have one image where they show a band pass. Are you familiar with band passes?
Yes.
It's essentially what you just described.
Yeah. It's
in terms of the logic gates, right? You either have 0 or 1 as the output.
Right.
Band passes, there's a gradient, right? It goes up and then back down again. So they do have one example of that where they have this purple Pac-Man that kind of modulates those signals. And so might that be applied to cancer cells? That's an interesting avenue.
Yeah.
In other words, the main takeaway is that the cancer cells die. I mean, you're saying you have dishonored yourself in your organism, and the cancer cells like, I don't deserve to live. And there you go, apoptosis.
Okay.
Right. But I think in conclusion, this is a really good example of bioengineering in terms of its design, its construction, its logic, and also an application as well.
Yeah.
So protein computing, I think, is a really interesting field that you and listeners should be looking forward to learn more about in the future.
Yeah,
Great
pretty
job,
interesting.
Al.
Appreciate it. Great.
All right, let's end it there. Thank you all for listening. And yeah, leave comments if you have questions.
Join us next week.
Yeah.